Hi class, this is your instructor, Scholar Huff. We're in Chapter 40, Plant Reproduction. The remarkable evolutionary success of flowering plants can be linked to their novel reproductive strategies. In this chapter, we will explore the reproductive strategies of angiosperms and how their unique features, flowers and fruits, have contributed to their success. This is, in part, a story of coevolution between plants and animals, like the adaptations in both the orchid and the wasp that feed on it in the picture you see here. That ensures greater genetic diversity by dispersing plant gametes widely. Once fertilization occurs, a series of developmental events coordinated with environmental inputs often leads to a waiting period and then germination and maturation of the next generation. In a stable environment, however, there are advantages to maintaining status quo genetically. Asexual reproduction is a strategy that produces cloned individuals, an unusual twist to sexual reproduction in some flowering plants. That is that senescence or aging and death of the parent plant immediately follows reproduction. In Chapter 30, it was noted that angiosperms represent an evolutionary innovation with their production of flowers and fruits. And here we begin with a vegetative plant and follow the developmental progression resulting in the elaborate structures associated with flowering. Recall that plants exhibit a haplodiplontic life cycle, often described as alternation of generations. A multicellular sporophyte generation produces a haploid spores by meiosis, and a multicellular gametophyte generation produces haploid gametes by mitosis. Two gametes fuse in fertilization to form a zygote that grows into a new sporophyte by mitosis. Plants go through developmental changes leading to reproductive maturity just as many animals do. This shift from juvenile to adult development is seen in the metamorphosis of a tadpole to an adult frog or a caterpillar to a butterfly that can then reproduce. Plants undergo a similar metamorphosis that leads to the production of a flower. Unlike the juvenile frog, which loses its tail, plant meristems keep adding structures to existing ones. Carefully regulated processes determine when and where flowers will form. Moreover, plants must often gain competence to respond to internal or external signals regulating flowering. Once plants are competent to reproduce, a combination of factors including light, temperature, or both promotive and inhibitory internal signals determines when a flower is produced. These signals turn on genes that specify formation of the floral organs. Sepals, petals, stamens, and carpels. Once cells have instructions to become a specific floral organ, yet another development cascade leads to the three-dimensional construction of flower parts. Most seedling plants are incapable of producing a flower, even if all the environmental cues are optimal. Internal developmental changes allow plants to obtain competence to respond to external or internal signals, or both, that trigger flower formation. This transition is referred to as phase change. Phase change can be morphologically obvious or very subtle. Take a look at an oak tree in winter. Leaves will still be clinging to lower branches until spring when new buds push them off, but leaves on the upper branches will have fallen earlier. These lower branches were initiated by a juvenile meristem. The fact that they did not respond to the environmental cues and drop their leaves indicates that they are juvenile branches and have not made a phase change. Although the lower branches are older, their juvenile state was established when they were initiated and will not change. Ivy also has distinct juvenile and adult phases of growth. Stem tissue produced by a juvenile meristem initiates adventitious roots, clinging to walls. If you look at very old brick buildings covered with ivy, you will notice that the uppermost branches are falling off because they have transitioned to the adult phase of growth and have lost their ability to produce adventitious roots. It is important to note that even though a plant has reached the adult stage of development, it may not produce reproductive structures. Other factors may be necessary to trigger flowering. Generally, it is easier to get a plant to revert from adult to juvenile state than to induce phase change experimentally. Applications of the plant hormone gibberellin and severe pruning can cause reversion. In the latter case, new vegetative growth occurs as well as when certain shrubs such as rhododendrons are cut back and put out lush growth in response. Four genetically controlled pathways regulate flowering. 1. The light-dependent pathway. 2. The temperature-dependent pathway. 3. The gibberellin-dependent pathway. And 4. The autonomous pathway. There is considerable variation in how flowering is influenced by environmental factors. Plants can rely primarily on one pathway, but all four pathways can be present. Flowering requires much energy accumulated via photosynthesis. Thus, all plants require light for flowering, but this is distinct from the photoperiodic or light-dependent flowering pathway. Aspects of growth and development in most plants are key to changes in the proportion of light to dark in the daily 24-hour cycle, or day length. Sensitivity to the photoperiod provides a mechanism for plants to respond to seasonal changes in the relative length of day and night. Day length changes with seasons. The farther a region is from the equator, the greater the variation in day length between winter and summer. Using light as a cue permits plants to flower when abiotic environmental conditions are optimal, pollinators are available, and competition for resources with other plants may be less. For example, the spring herbaceous plants termed ephemerals flower in the woods before the tree canopy leaves out, blocking the sunlight necessary for photosynthesis. The flowering responses of plants to day length fall into several basic categories. In short day plants, flowering begins when days become shorter than a critical length. Conversely, in long day plants, flowering begins when the day length is longer than a critical length. Day neutral plants flower when mature, regardless of day, as long as they have enough light for normal growth. These include snapdragons, roses, and many tropical plants. Still, other plants, including ivy, have two critical photoperiods. These species will not flower if the days are too long, but they also will not flower if the days are too short. Although we use terms like long day and short day, it is actually the amount of darkness that determines whether a plant will flower. So an obligate long day species is actually responding to short nights, and a short day plant is responding to long nights. In obligate long day plants, flowering occurs when the duration of darkness is less than a critical night length for that species. In obligate short day plants, flowering occurs when the duration of darkness exceeds a critical night length for the species. Some long or short day plants do not have an absolute dependence on light duration, but will flower more rapidly or slowly depending upon the length of day. These plants, which rely on other flowering pathways as well, are called facultative long or short day plants. The garden pea is an example of a facultative long day plant. At middle latitudes, most long day plants flower in the spring and early summer. Examples of such plants include clover, irises, lettuce, spinach, hollyhocks. Short day plants usually flower in late summer and fall. These include chrysanthemums, goldenrods, poinsettias, soybeans, and many weeds such as ragweed. Commercial plant growers use these responses to day length to bring plants into flower at specific times. For example, photoperiod is manipulated in greenhouses so that poinsettias flower at just the right time for the winter holidays. The geographic distribution of certain plants may be determined by their flowering responses to day length. Photoperiod is perceived by several different forms of phytochrome and also by blue light sensitive molecule called cytochrome. The conformational change in phytochrome or cryptochrome light receptor molecules triggers a cascade of events that leads to the production of a flower. There is a link between light and the circadian rhythm regulated by an internal clock that facilitates or inhibits flowering. We do not know all of the steps between light reception and flower production, but control of mechanisms appear to be quite complex. Plants also use temperature as an environmental cue to ensure they flower at optimal times. Vernalization is the requirement for a period of chilling of seeds or shoots before flowering can occur. Plants with a vernalization requirement flower after, not during, a cold winter, enhancing reproductive success. The process of vernalization was discovered in the 1930s by Ukrainian scientist T.D. Lysenko while trying to solve the problem of the rotting winter wheat in the fields, because winter wheat would not flower without a period of chilling. Lysenko chilled the seeds, then planted them in the ground in the spring. The seeds successfully sprouted, grew, and produced grain. Although this discovery was scientifically significant, Lysenko erroneously concluded that he had converted one species, winter wheat, to another, spring wheat, by simply altering the environment. Lysenko's point of view was consistent with the communist philosophy of the time, which held that people could easily manipulate nature to increase production. This had unfortunate political ramifications, including mistreatment of legitimate geneticists in the Soviet Union. In addition, geneticists and Darwinian evolution were suspect in the Soviet Union until the mid-1960s. The gibberellin-dependent pathway. Decreased levels of gibberellins have been shown to delay flowering in some species. This implies that gibberellin signal transduction pathway promotes flowering. The autonomous pathway to flowering does not depend on external cues except for basic nutrition, which is necessary for all pathways. A balance between floral promoting and inhibiting signals may regulate when flowering occurs in the autonomous pathway and the other pathways as well. So the ABC model explains how three classes of floral organ identity genes can specify four distinct organ types. So class A genes alone, sepals, class A and B genes together, petals, class B and C genes together, stamens, and class C genes alone, carpels. When any one class is missing, aberrant floral organs can occur in predictable positions. The complex and elegant process that gives rise to the flower is often compared with metamorphosis in animals. It is indeed a metamorphosis, but the subtle shift from mitosis to meiosis in the megaspore mother cell that leads to the development of a haploid gamete-producing gametophyte is perhaps even more critical. The same can be said for pollen formation in the anther of the stamen. The flower not only houses the haploid generations that will produce gametes, but also functions to increase the probability that male and female gametes from different sporophytes will unite, except in the case where they actually do the opposite. The evolution of the angiosperms was what you were taught back in chapter 30. The diversity of angiosperms is partly due to the evolution of a great variety of floral phenotypes that may enhance the effectiveness of pollination. As mentioned in the prior section, floral organs are thought to have evolved from leaves. In some early angiosperms, these organs maintain the spiral development pattern found in leaves. The trend has been toward four distinct whorls of parts. A complete flower has four whorls: the calyx, corolla, androecium, and the gynoecium or gynoecium. An incomplete flower lacks one or more of those whorls. Flower morphology, and this is a bit of review. In both complete and incomplete flowers, the calyx usually constitutes the outermost whorl. It consists of flattened appendages called sepals, which protect the flower in the bud. Modified leaves called petals make up the next whorl, which is called the corolla. Petals may be individual or fused, and they are often brightly colored and shaped in ways to attract pollinators. Although these two outer whorls of floral organs are not involved directly in gamete production or fertilization, they can enhance reproductive success. Male structures. The next whorl is the androecium, which is a collective term for all the stamens of a flower. Stamens are specialized structures that bear the angiosperm microsporangia. Most angiosperms have stamens with filaments that are slender and often thread-like, which are tipped with four microsporangia at the apex and a swollen portion called the anther. The microsporangia in the anther make spores that grow into pollen grains. The androecium, stamen, and pollen are all considered male structures because pollen makes sperm. Female structures. The gynoecium is a collective term for all parts of a flower. That is, parts of the flower involved with forming an egg. In most flowers, the gynoecium, which is unique to angiosperms, consist of a single carpel or two or more fused carpels. Single or fused carpels are often referred to as simple or compound pistils, respectively. Most flowers with which we are familiar, for example, those of tomatoes and oranges, have a compound pistil. Other, less specialized flowers, for example, buttercups and stonecups, may have several to many separate simple pistils, each formed from a single carpel. Ovules. Ovules, which develop into seeds, are produced in the pistil's swollen lower portion, the ovary, which usually narrows at the top into a slender neck-like style with a pollen-receptive stigma at its apex. Sometimes the stigma is divided, with a number of stigma branches indicating how many carpels compose the particular pistil. Carpels are essentially rolled floral leaves with ovules along the margins. It is possible that the first carpels were leaf blades that folded longitudinally. The edges of the leaf, which had hairs, did not actually fuse until the fruit developed, but the hairs interlocked and were receptive to pollen. In the course of evolution, evidence indicates that the hairs became localized into stigma and a style was formed, and the fusing of the carpel margins ultimately resulted in a pistil. In modern flowering plants, the carpels have become highly modified and are not visually distinguishable from one another unless the pistil is cut open. Two major evolutionary trends led to the wide diversity of modern flowering plants. First, floral parts have grouped together or fused, and second, floral parts have been lost or reduced. In the more advanced angiosperms, the number of parts in each whorl has often been reduced from many to few. The spiral patterns of attachment of all floral parts in primitive angiosperms have, in the course of evolution, given way to a single whorl at each level. The central axis of many flowers has shortened, and the whorls are close to one another. In some evolutionary lines, the members of one or more whorls have fused with one another, sometimes joining into a tube. In other kinds of flowering plants, different whorls may be fused together. Whole whorls may even be lost from the flower, which may lack sepals, petals, stamens, carpels, or various combinations of these structures. And modifications often relate to pollination mechanisms, and in plants such as the grasses, wind has replaced animals for pollen dispersal. Other trends in floral evolution have affected the symmetry of a flower. Primitive flowers, such as those of buttercups, are radially symmetrical. That is, one could draw a line through anywhere through the center and have roughly two equal halves. Flowers of many advanced groups are bilaterally symmetrical. They are divisible into two equal parts along only a single plane. Examples of such flowers are snapdragons, mints, and orchids. Bilaterally symmetrical flowers are also common among violets and peas. In these groups, the flowers often have advanced and highly precise pollination systems. Although much floral diversity is a result of natural selection related to pollination, it is important to recognize the effect breeding, artificial selection has had on flower morphology. Humans have selected for practical or aesthetic traits that have little adaptive value to species in the wild. For example, corn or maize has been bred to satisfy the human palate. Human intervention ensures the reproductive success of each generation. However, in a natural setting, modern corn would not have the same protection from herbivores as its ancestors. the fruit dispersal mechanism will be quite different. Reproductive success depends on uniting gametes found in the embryo sacs and pollen grains of flowers. As you learned earlier, plant sexual life cycles are characterized by an alternation of generations in which diploid sporophyte generation gives rise to a haploid gametophyte generation. In angiosperms, the gametophyte generation is very small and is completely enclosed within tissues of the parent sporophyte. The gametophytes, or microgametophytes, are pollen grains. The female gametophyte, or megagametophyte, is the embryo sac. Pollen grains and the embryo sac both are produced in separate, specialized structures of the angiosperm flower. Like animals, angiosperms have separate structures for producing male and female gametes, but the reproductive organs of angiosperms are different from those of the vertebrate animals we are most familiar with. First, both male and female structures usually occur together in the same individual flower. Second, angiosperm reproductive structures are not permanent parts of the adult sporophyte. Angiosperm flowers and the reproductive organs develop seasonally at times of the year most favorable for pollination. In some cases, reproductive structures are produced only once and the sporophyte dies. And as you learned in the first section of this chapter, the germline in angiosperms is not set aside early on, but forms quite late during phase change. Anthers contain 4 microsporangia, which produce microspore mother cells. Microspore mother cells produce microspores through meiotic division. The microspores, through mitosis and wall differentiation, become pollen. Inside each pollen grain is a generative cell that later divides to produce 2 sperm cells. Pollen grain shapes are specialized for the specific flower species. Fertilization requires that the pollen grain grow a tube that penetrates the style until it encounters the ovary. Most pollen grains have a furrow or pore from which this pollen tube emerges, and some grains have 3 furrows. Eggs develop in the ovules of the angiosperm flower. Within each ovule is a megaspore mother cell. Just as in pollen production, the megaspore mother cell undergoes meiosis to produce 4 haploid megaspores. In most plants, however, only one of these megaspores survives. The rest are absorbed by the ovule. The lone remaining megaspore enlarges and undergoes repeated mitotic divisions to produce 8 haploid nuclei that are enclosed within a 7-celled embryo sac. The embryo sac is the female gametophyte, and one of those 7 cells is an egg. Within the embryo sac, the 8 nuclei are arranged in precise positions. One nucleus is located near the opening of the embryo sac in the egg cell. 2 others are located together in a single cell in the middle of the embryo sac. These are called polar nuclei. 2 more nuclei are contained in the individual cells called synergids that flank the egg. The other 3 nuclei reside in cells called the antipodals, located at the end of the embryo sac, opposite the egg cell. The first step in uniting sperm cells made by the pollen grain to the egg and polar nuclei is the germination of pollen on the stigma of the carpel. Pollination and fertilization are different events involving different processes. Pollination is the process by which pollen is placed on the stigma. Fertilization is the unification of haploid gametes to produce a diploid zygote. Pollen from one plant's flowers can be carried to the flowers of another plant by air currents or by animals. In some cases, pollen from a flower's anthers can pollinate the same flower stigma in a process called self-pollination. This is a form of inbreeding. When the pollen from another of one flower pollinates the stigma of flowers on a different plant, the process is termed cross-pollination or outcrossing. So again, when the pollen from the anther of one flower pollinates the stigma of flowers on a different plant, it is cross-pollination and outcrossing. Pollination in angiosperms does not involve direct contact between pollen grain and the ovule. When pollen reaches the stigma, it germinates and the pollen tube grows down, carrying the sperm nuclei to the embryo sac. After double fertilization takes place, development of the embryo and endosperm begins. The seed matures within the ripening fruit. Eventually, the germination of the seed allows the embryo to grow into a new sporophyte. Successful pollination in many angiosperms depends on the regular attraction of pollinators, such as insects, birds, and other animals, which transfer pollen between plants of the same species. When animals disperse pollen, they perform the same function for flowering plants that do not do for themselves when they actively search out mates. The relationship between plant and pollinator can be quite intricate. Mutations in either partner can block reproduction. If a plant flowers at the wrong time, the pollinator may not be available. If the morphology of the flower or pollinator is altered, the result may be physical barriers to pollination. Clearly, floral morphology has co-evolved with pollinators, and the result is a much more complex and diverse morphology, going beyond simple initiation and development of 4 distinct worlds of organs. Outcrossing, as we have stressed, is critically important for the adaptation and evolution of all eukaryotic organisms, with a few exceptions. Often flowers contain both stamens and pistils, which increases the likelihood of self-pollination. One strategy to promote outcrossing, therefore, is to separate stamens and pistils. Another strategy involves self-incompatibility that prevents self-fertilization. In a number of species, for example, willows and some mulberries, staminate and pistillate flowers may occur on separate plants. Such plants, which produce only ovules or only pollen, are called dioecious, meaning 2 houses. These plants clearly cannot self-pollinate and must rely exclusively on outcrossing. In other kinds of plants, such as oaks, birches, corn, and pumpkins, separate male and female flowers may both be produced from the same plant. Such plants are called monoecious. Monoecious means 1 house. In monoecious plants, the separation of pistillate and staminate flowers, which mature at different times, greatly enhances the probability of outcrossing. Even if, as usually is the case, functional stamens and pistils are both present in each flower of a particular plant species, these organs may reach maturity at different times. Plants in which this occurs is called dichogamous. If the stamens mature first, shedding their pollen before stigmas are receptive, the flower is effectively staminate at that time. Once stamens have finished shedding pollen, the stigma or stigmas may become receptive and flower may become essentially pistillate. This separation in time has the same effect as if individuals were dioecious. The outcrossing rate is thereby significantly increased. Many flowers are structured so that the stamens and stigmas do not come in contact with each other. With this arrangement, the natural tendency for pollen to be transferred to the stigma of another flower rather than this of its own flower is thereby promoting outcrossing. Even when a flower's stamens and stigmas mature at the same time, genetic self-incompatibility, which is widespread in flowering plants, increases outcrossing. Self-incompatibility results when the pollen and the stigma recognize each other as being genetically related, and the pollen tube growth is blocked. Early seed plants were pollinated passively by the action of the wind. As in present-day conifers, great quantities of pollen were shed and blown about, occasionally reaching the vicinity of the ovules of the same species. Individual plants of any wind-pollinated species must grow relatively close to one another for such a system to operate efficiently. Otherwise, the chance that any pollen will arrive at an appropriate destination is very small. The vast majority of wind-blown pollen travels less than 100 meters. This short distance is significant compared with the long distances pollen is carried by certain insects, birds, and other animals. The spreading of pollen from plant to plant by pollinators visiting flowers of an angiosperm species has played an important role in the evolutionary success of the group. It now seems clear that the earliest angiosperms, and perhaps their ancestors, also were insect pollinated, and the coevolution of insects and plants has been important for both groups for over 100 million years. Such interactions have also been important in bringing about increased floral specialization. As flowers become increasingly specialized, so do the relationships with particular groups of insects and other plants. Among insect-pollinated angiosperms, the most numerous groups are those pollinated by bees. Like most insects, bees initially locate sources of food by odor, then orient themselves on the flower or a group of flowers by shape, color, and texture. Flowers that bees characteristically visit are often blue or yellow because bees detect color from yellow through ultraviolet. Carotenoids are the yellow to orange pigments, responsible for the colors of many flowers, including sunflowers and mustard. Carotenoids reflect both in yellow range and the ultraviolet range, and the resulting mixture is a distinctive color called bees purple. Such yellow flowers may be marked with stripes or lines of dots that form bullseye or landing strip features that are only visible in ultraviolet light. Except for a few hundred species of social and semi-social bees, about 1,000 species that are parasitic in nests of other bees. The great majority of bees, at least 18,000 species, are solitary. Solitary bees in temperate regions are characteristically produce only a single generation in the course of a year. Often they are active as adults for as little as a few weeks a year. Solitary bees often use the flowers of a particular group of plants almost exclusively as a source of larval food. The highly constant relationships of bees with those flowers may lead to modifications over time in both flowers and bees. For example, the time of day when the flowers open may correlate with the time the bees appear. The mouthparts of bees may become elongated in relation to the tubular flowers. Or the bees' pollen collecting apparatus may be adapted to the anthers of the plants that they normally visit. When such a relationship is established, they provide both an efficient mechanism of pollination for the flowers and a constant source of food for the bees that specialize on them. Other than bees, among flower-visiting insects, a few groups are especially prominent. Flowers such as phlox, which are visited regularly by butterflies, often have flat landing platforms on which butterflies perch. They also tend to have long, slender floral tubes filled with nectar that is accessible by the long coiled proboscis, characteristic of Lepidoptera, the order of insects that includes butterflies and moths. Flowers such as jimson weed, evening primrose, and others regularly visited by moths are often white and some other pale color. They also tend to be heavily scented, making the flowers easy to locate at night. Skunk cabbage produces an odor that many people find unpleasant, but that seems to attract a variety of flies that serve as pollinators. Several interesting groups of plants are regularly visited and pollinated by birds, especially hummingbirds of North and South America and the sunbirds of Africa. Such plants must produce large amounts of nectar because birds will not continue to visit flowers if they do not find enough food to maintain themselves. But flowers producing large amounts of nectar have no advantage in being visited by insects because an insect could obtain its energy requirements at a single flower and would not cross-pollinate the flower. How do these different selective forces balance in flowers that are specialized for hummingbirds and sunbirds? The answer involves the coevolution of flower color. Unlike the yellow flowers discussed earlier with bees, insects do not see longer wavelengths associated with red flowers. However, red is very conspicuous to birds. Consequently, even though the flowers produce abundant supplies of nectar and attract hummingbirds, insects tend to pass by them. Thus, the red color signals to birds the presence of abundant nectar and makes that nectar inconspicuous as possible to insects. Red is also seen again in fruits that are dispersed by birds. Other animals, including bats, small rodents may aid in pollination. The signals here are species-specific. As an example, the saguaro cactus of the Sonoran Desert is pollinated by bats that feed on the nectar at night, as well as by birds and insects. The saguaro blossoms are large, white flowers that further attract bats by opening and emitting fragrance at night. A number of groups of angiosperms are wind-pollinated, a characteristic of early seed plants. Among these groups are oaks, birches, cottonwoods, grasses, sedges, and nettles. The flowers of these plants are small, greenish, and odorless. Their corollas are reduced or absent, as you see here and here. Such flowers are often grouped together in fairly large numbers and may hang down in tassels that wave about. In the wind to shed pollen freely. Many wind-pollinated plants have stamen and carpellet containing flowers separated between individuals or physically separated on a single individual. Maize, that's corn, you all, is a good example. With pollen-producing tassels at the top of the plant and the axillary shoots with female flowers lower down. Separation of pollen-producing and ovule-bearing flowers is a strategy that greatly promotes outcrossing, since pollen from one flower must land on a different flower for fertilization to have the chance of occurring. Some wind-pollinated plants, especially trees and shrubs, flower in the spring before their leaves can interfere with the dispersal of wind-borne pollen. Wind-pollinated species do not depend on the presence of a pollinator for species survival, which may be another survival advantage. Thus far, we have considered examples of pollination that tend to lead to outcrossing, which is highly advantageous for plants and eukaryotic organisms generally. Nevertheless, self-pollination also occurs among angiosperms, particularly in temperate regions. Most self-pollinated plants have small, relatively inconspicuous flowers that shed pollen directly onto the stigma, sometimes even before the bud opens. You might logically ask why many self-pollinated plant species have survived if outcrossing is as important genetically for plants as it is for animals. Two reasons have been proposed. Self-pollination is favored in environments where pollinators are scarce, and offspring are more uniform and probably better adapted to their environment, which is favored in stable locations. And remember, meiosis is involved, so recombination still takes place, and therefore offspring will not be identical to the parent. However, such progenies may contain high proportions of individuals well adapted to particular habitats. Fertilization in angiosperms is a process in which two sperm cells are required for a unique process called double fertilization. Double fertilization results in two key developments. One, the formation of a zygote that grows into an embryo, and two, the formation of a nutrient substance called endosperm that nourishes the embryo. Once a pollen grain has been spread by wind, by animals, or through self-pollination, it adheres to the sticky, sugary substance that covers the stigma, and it begins to grow a pollen tube that pierces the style. The pollen tube grows until it reaches the ovule in the ovary. Meanwhile, the generative cell within the pollen grain tube cell divides to form two sperm cells. During growth, the pollen tube is continuously perceiving and secreting chemical signals. When it reaches the ovary, the pollen tube is attracted by chemicals to release one of the synergids flanking the cell. When the pollen tube and the synergid make contact, the synergid disintegrates and the tip of the pollen tube bursts. The two sperm cells are released into the cell wall, free cleft between the egg and central cell. One of the sperm cells fuses with the egg cell, forming a zygote. The other sperm cell fuses with the two polar nuclei located at the center of the embryo sac, forming the triploid primary endosperm nucleus. The primary endosperm nucleus eventually develops into the endosperm that supplies nutrients to the developing embryo. Once fertilization is complete, the embryo develops as its cells divide numerous times. Meanwhile, the protective tissues enclose the embryo, resulting in formation of the seed. The seed, in turn, is enclosed in another structure called the fruit. Evolution of these typical angiosperm structures allows seeds to be dispersed over long distances to ensure genetic variability. Embryo development begins once the egg is fertilized. As described briefly in Chapter 30, the growing pollen tube from a pollen grain enters the angiosperm embryo sac through one of the synergids, releasing two sperm cells. One sperm cell fertilizes the central cell with its polar nuclei, and the resulting cell division produces a nutrient source, the endosperm, for the embryo. The other sperm cell fertilizes the egg to produce the zygote, and the cell division soon follows, creating the embryo. The first division of the zygote, the fertilized egg, in a flowering plant is asymmetrical and generates cells with two different fates. The smaller apical cell, which is destined to become the embryo, begins to divide repeatedly in different planes, forming a ball of cells. The larger basal cell divides repeatedly, forming an elongated structure called a suspensor, which links the embryo to the nutrient tissue of the seed. The suspensor also provides a route for nutrients to reach the developing embryo. The root-shoot axis also forms at this time. Cells near the suspensor are destined to form a root, and those at the other end of the axis ultimately become a shoot. Three basic tissues, called primary tissues, differentiate while the plant embryo is still a ball of cells, called the globular stage. No cell movements are involved in plant embryo development. The protoderm consists of the outermost cells in the plant embryo and will become the dermal tissue. These cells almost always divide with their cell plate perpendicular to the body surface, thus perpetuating the single cell layer of outer cells. The single outer layer of cells. Dermal tissue protects the plant from desiccation and attack by pathogens. Stomata that open and close and facilitate gas exchange and minimize water loss are derived from dermal tissue. A ground meristem gives rise to the bulk of the embryonic interior, consisting of ground tissue, cells that eventually function in food and water storage. And finally, the procambium at the core of the embryo will form the future vascular tissue, which is responsible for water and nutrient transport. And this is what I just provided to you all there. The globular stage gives rise to a heart-shaped embryo with two bulges and one group of angiosperms, the eudicots, and a ball with a bulge on a single side in another group, the monocots. These bulges are cotyledons, or the first leaves, and are produced by the embryonic cells and not by the shoot apical meristem that begins forming during the globular stage. This process is called morphogenesis, the development of form, and results from changes in planes and rates of cell division. While the embryo is developing, three other critical events are occurring in angiosperms. 1, the development of food supply. 2, the development of the seed coat. And 3, development of the fruit surrounding the seed. Nutritional reserves support the embryo during germination while it gains photosynthetic capacity. As discussed earlier, endosperm formed during double fertilization provides nutrition for the angiosperm embryo. The seed coat is a result of differentiate of ovule tissue to form a hard protective covering around the embryo, and after the seed coat is formed, the seed enters the dormant phase. Throughout embryogenesis, starch, lipids, and proteins are synthesized. The seed storage proteins are so abundant that the genes coding for them were the first cloning targets for plant molecular biologists. The sporophyte transfers nutrients via the suspensor to the angiosperm in angiosperms. This happens concurrently with the development of the endosperm, which is only present in angiosperms. Endosperm formation may be extensive or minimal. Endosperm in coconut includes the milk, a liquid. In corn, the endosperm is solid. In popping popcorn, it expands with heat to form the white edible part of popped corn. In peas and beans, the endosperm is used up during embryo development, and nutrients are stored in thick, fleshy cotyledons, or seed leaves. Because photosynthetic machinery is built in response to light, it is critical that seeds have stored nutrients to aid in germination until the growing sporophyte can photosynthesize. A seed buried too deeply in the soil will use up all its reserves in cellular respiration before reaching the surface and sunlight. When conditions are satisfactory, the embryo emerges from its previously desiccated state, utilizes food reserves, and resumes growth. Although germination is a process characterized by several stages, it is often defined as the emergence of the radicle, the first root, through the seed coat. Germination begins when a seed absorbs water, and its metabolism resumes. The amount of water a seed can absorb is phenomenal, and the osmotic pressure creates a force strong enough to break the seed coat. At this point, it is important that oxygen be available to the developing embryo because plants, like animals, require oxygen for cellular respiration. Few plants produce seeds that germinate successfully underwater, although some, such as rice, have evolved a tolerance to anaerobic conditions. Even though a dormant seed may have imbibed a full supply of water and may be respiring, synthesizing proteins and RNA, and apparently carrying on normal metabolism, it may fail to germinate without an additional signal from the environment. The signal may be light of the correct wavelength and intensity, a series of cold days, or simply the passage of time at temperatures appropriate for germination. The seeds of many plants will not germinate unless they have been stratified, held for periods of time at low temperatures. This phenomenon prevents the seeds of plants that grow in seasonally cold areas from germinating until they have passed the winter, thus protecting their tender seedlings from harsh, cold conditions. Germination can occur over a wide temperature range, although certain species may have relatively narrow optimum ranges. Some seeds will not germinate even under the best conditions. In some species, a significant fraction of seasoned seeds remain dormant for an indeterminate length of time, providing a gene pool of great evolutionary significance to the future plant population. The presence of ungerminated seeds in the soil of an area is referred to as the seed bank. Germination and early seedling growth require the utilization of metabolic reserves stored as starch in the ameloplasts and protein bodies. Depending on the kind of plant, these reserves may be stored in the embryo or in the endosperm. The scutellum in kernels of cereal grains, the single cotyledon, is modified into the structure, which transfers nutrients from the endosperm to the embryo. And this shows germination and early growth. On the left-hand side, it shows what occurs in the eudicots. This is a common bean. And on the right-hand side, with B, that shows what occurs in the monocots. That is maize. Self-pollination may result in heterozygous individuals producing offspring that exhibit deleterious homozygous recessive conditions, but asexual reproduction results in genetically identical individuals because only mitotic cell divisions occur. In the absence of meiosis, individuals that are highly adapted to a relatively unchanging environment persist for the same reason that self-pollination is favored. Should conditions change dramatically, there will be less variation in the population for natural selection to act on, and the species may be less likely to survive. Asexual reproduction is far more common in harsh or marginal environments, where there is little leeway for variation. For example, a greater proportion of asexual plants occurs in the Arctic than in temperate regions. Asexual reproduction is also used in agriculture and horticulture to propagate a particularly desirable plant with traits that would be altered by sexual reproduction or even by self-pollination. Most roses and potatoes, for example, are vegetatively asexually propagated. In certain plants, including some citrus and certain grasses, such as Kentucky bluegrass and dandelions, the embryos in the seeds may be produced asexually in the carpel. This kind of asexual reproduction is known as apomixis. A seed produced in this way will give rise to an individual that is genetically identical to its parent. Although these plants reproduce by cloning diploid cells in the ovule, they also gain the advantage of seed dispersal, an adaptation usually associated with sexual reproduction. In a very common form of asexual reproduction called vegetative reproduction, new plant individuals are simply cloned from parts of adults. Runners or stolons. Some plants reproduce by means of runners, also called stolons. Long, slender stems that grow along the surface of the soil. In cultivated strawberry, for example, leaves, flowers, and roots are produced at every other node on the runner. Just beyond the second node, the tip of the runner turns up and becomes thickened. This thickened portion produces the first adventitious roots, then a new shoot that continues the runner. Rhizomes, horizontal underground stems, are also important reproductive structures, particularly in grasses and sedges. Rhizomes invade areas near the plant at each node can give rise to a new flowering shoot. The noxious character of many weeds results from this type of growth pattern, and many garden plants, such as irises, are propagated almost entirely from rhizomes. Corms and bulbs are vertical underground stems. Tubers are also stems specialized for storage and reproduction. Tubers are the terminal storage portion of a rhizome. Potatoes are propagated artificially from tuber segments, each with one or more eyes. The eye or the seed pieces of potato give rise to the new plant. Suckers, the roots of some plants, for example, cherry, apple, raspberry, blackberry, produce suckers or sprouts which give rise to new plants. Commercial varieties of banana produce only sterile seeds and are propagated instead by suckers that develop from buds on underground stems. When the root of a dandelion is broken, as it may be if one attempts to pull it from the ground, each root fragment may give rise to a new plant. And you're seeing before yourself plantlets, an unusual form of vegetative reproduction. So adventitious plantlets, in a few species, even the leaves are reproductive. So this is the common name called mother of thousands, or the maternity plant. So the common names of this plant are based on the fact that numerous plantlets arise from meristematic tissue located in notches on the leaves. This plant is ordinarily propagated by means of these small plants, which when they mature, drop to the soil and take root. So whole plants can also be cloned by regenerating plant cells or tissues on nutrient medium with hormones. Individual cells can be isolated from tissues with enzymes that break down cell walls, and what's left behind is a protoplast, the plant cell enclosed by only a plasma membrane. Many but not all cell types in plants maintain the ability to generate organs or entire organisms in culture. And the cells divide in culture, regenerating cell walls to form an undifferentiated mass of cells called a callus. So whole plants can be produced in culture from a callus. So tissue culture has many agricultural and horticultural applications. So virus-free raspberries and sugarcane can be propagated by culturing meristems, which are generally free of viruses, even in an infected plant. As with other forms of asexual reproduction, genetically identical individuals can be propagated. And finally, plant lifespans. Once established, plants live for highly variable periods of time, depending on species. Lifespan may or may not correlate with reproductive strategies. Woody plants, which have an extensive secondary growth, nearly always live longer than herbaceous plants, which have limited or no secondary growth. Bristlecone pine, for example, can live over 4,800 years. Some herbaceous plants send new stems above ground every year, producing them from woody underground structures. Others germinate and grow, flowering just once before they die. Shorter-lived plants rarely become very woody because there is not enough time for secondary tissues to accumulate. So depending on their life cycles, herbaceous plants may be annual, biennial, or perennial, whereas woody plants are generally perennial. Determining the lifespan is even more complicated for clonally reproducing organisms. Aspen trees form huge clones from asexual reproduction of the roots. Collectively, an aspen clone may be the largest organism on Earth. Other asexually reproducing plants may cover less territory but live for thousands of years. Creosote bushes in the Mojave Desert have been identified that are as much as 12,000 years old. The creosote bushes. Perennials. Perennial plants continue to grow year after year and may be herbaceous, as are many woodland, wetland, and prairie wildflowers, or woody as trees or shrubs. The majority of vascular plant species are perennials. Perennial plants in general are able to flower and produce seeds and fruit for an indefinite number of growing seasons. Herbaceous perennials rarely experience secondary growth in their stems. The stems die each year after a period of relatively rapid growth and food accumulation. Food is often stored in the plant's roots or underground stems, which can become quite large in comparison to their less substantial above-ground counterparts. Trees and shrubs generally flower repeatedly, but there are exceptions. Bamboo lives for many seasons as a non-reproducing plant, but senesces and dies after flowering. The same is true for at least one tropical tree, which achieves great heights before flowering and senescing. Considering the tremendous amount of energy that goes into the growth of a tree, this particular reproductive strategy is quite curious. Trees and shrubs that are either deciduous, with all leaves falling at one time of the year, and plants remaining bare for a period, or evergreen, with leaves dropping throughout the year, and the plants never appearing completely bare. In northern temperate regions, conifers are the most familiar evergreens, but in tropical and subtropical regions, most angiosperms are evergreen except where seasonal drought occurs. In these areas, many angiosperms are deciduous, losing their leaves during the dry season, thus conserving water. So you're seeing a tree here, the giant redwood, which can live 2,000 years or more. So before I get to the end, there is a couple more parts. Annual plants grow, flower, and form fruits and seeds within one growing season and die when the process is complete. Many crop plants, including corn, wheat, and soybeans, are annuals. So annuals generally grow rapidly under favorable conditions and in proportion to the availability of water and nutrients. The lateral meristems of some annuals, such as sunflowers or giant ragweed, produce some secondary tissues for support, but most annuals are entirely herbaceous. Annuals typically die after flowering once. The developing flowers or embryos use hormonal signaling to reallocate nutrients, so the parent plant literally starves to death. This can be demonstrated by comparing a population of bean plants in which the beans are continually picked with a population in which beans are left on the plant. The frequently picked population will continue to grow and yield beans much longer than the untouched population. The process that leads to death of a plant is called senescence. Biennial plants, which are much less common than annuals, have life cycles that take 2 years to complete. During the first year, biennials store the products of photosynthesis in underground storage organs. During the second year of life, flowering stems are produced, using energy stored in the underground parts of the plant. Certain crop plants, including carrots, cabbage, and beetles, are biennials, but these plants generally are harvested for food during their first season, before they flower. They are grown for their leaves, roots, not for fruits or seeds. Wild biennials include evening primroses, Queen Anne's lace, and mullein. Many plants that are considered biennials actually do not flower until they are 3 or more years old, but all biennial plants flower only once before they die. And these are desert annuals you're seeing here at A. This has been your instructor, Scholar Huff.